Hello everyone, my name is Silke Asche. I'm a postdoctoral program fellow at the NASA Goddard Space Flight Center. My research focuses on agnostic biosignature method development. My seminar is going to be titled Experimental Molecular Assembly Estimations for Space Exploration Using Gas Chromatography Mass Spectrometry. So in my research, we focus on recognizing life as we might not know it, and we want to be able to measure anomalies in molecular assemblages in unexplored environments. A method that we found quite interesting for this is molecular assembly, which has been proven to show differences in biological to abiotic samples using electrospray ionization mass spectrometry. However, there is a problem with this because electrospray ionization is an instrument that is incredibly powerful, but it's not spaceflight ready. So we went ahead and tried to find a way to do molecular assembly estimations using electron impact ionization, gas chromatography, mass spectrometry. And so we ran a bunch of laboratory tests of known standards, complex extraterrestrial and biological samples. And we found that we can do electron ionization, molecular assembly estimations, and that we can really show the differences in these sample types that we measured. And if you want to know more about this, I'm looking forward to tell you more in detail in the next seminar.